Okay. All right. Hi, everybody. I'm going to switch over to my slides really quickly and give you an idea of where we're going today. Let me do that.
And Liz, would you like to briefly introduce yourself and what
I
your lab
will. I
does?
absolutely will. It's on. Yeah, I've got a space for it. Right, so I'm going to introduce myself in a second. But before we got started, I just wanted to mention the sort of the context for this lecture. I know you're just starting out on the whole boot camp experience, but I wanted to say that this whole, all of CEMB is about mechanobiology, which is a very broad term for the study of force in living systems, right? So this can be how molecules or cells or tissues or organisms even generate force or how they respond to force. But what I'm going to talk about today is just a sliver of that, which is how they sense force. And we're going to really focus on this at the molecular level. And because of my background, I'm going to be focusing a lot on plant mechanoperception and mechanotransduction, but I will try to connect it to animal biology when I can. Okay, so here's today's plan. First, we'll do some introductions. I'll tell you a little bit about myself, and then I'm hoping, since there's just a few of you, to get like a really quick, have each of you introduce yourselves really quickly. And then we're going to talk about the lecture is divided into two sections, one on mechanotransduction and one really digging down into mechanosensitive ion channels. As I said, I'm going to stop sharing for a second so you can see my face. So as we said, my name is Liz Haswell. I'm a professor in the biology department. Although these days it feels like I'm a professor in my condo at WashU. And I just stepped down as research director for the CEMB. I've been at WashU for almost 13 years now. I came here from Caltech, where I did my postdoctoral research, and before that, I got a PhD in biochemistry at UC San Francisco. So my background and area of interest is biochemistry. I switched to plant work when I went to a postdoc, so prior to that, I worked on actually chromatin remodeling in yeast. So I have kind of a broader animal plant perspective, which you'll find out is actually really common among plant biologists to have moved in from other areas of research. And as I move farther into plant biology and into mechanobiology, I've been learning more and more both about plants and plant biology and more about physics. So you'll see sort of how those things come into play in my talk. Okay, so maybe I can get just a quick, well, maybe I'm seeing now there's so many of you, maybe this isn't such a great plan. So maybe I'll skip over the introductions, but do know that I care about you and who you are. And I would love for you to ask me questions. So please don't hesitate to interrupt me. And also I'll try to keep an eye up for hands raised. The other thing I want you to know is that I will be doing what's called, well, please ask questions. And I'm going to be doing a couple of what are called think, pair, share activities. And they sound cheesy, but they're kind of fun. I'm going to automatically sort you into a couple of breakout sessions. Depending on how our time goes, I'll be trying to do that a couple times during the lecture. And the idea there is that there's 3 steps. The first thing you do is you think about the answer to the question, which is going to be asking you to sort of integrate some of what I've presented, then pairing, or it'll probably be more than pairs. And then you talk about the answer to the question with your partners in your breakout group, and then you'll come back here and share your answers. So it's an opportunity for you to first think on your own and then have more of a discussion. Okay, any questions before we get going? Okay. All right, so this is no news to you, I'm sure, but all organisms sense and respond to mechanical force. And these forces can either be external, like gravity or touch, or they can be internal, like osmotic pressure and stress. And in fact, you're probably doing all of those right now as you're listening to me. Oops, this is like on some weird timer. um, you're sitting in your chair through the force of gravity. You can probably feel your chair touching your body where you're sitting on it. Osmotic pressure is keeping your blood volume high while you're sitting down, so you have enough blood going to your head to make your brain work. And if you get up and walk around, if you're at a standing desk, you're experiencing some stress or strain in your bones. So this is true of us, but it's also true of plants. And so just to give you a sense, I think many people, I know there are some plant biologists here, so you're all on board, but many people are not too clear on the idea that plants can sense and respond to much of anything, other than sometimes people think, oh, they sense light. But I want to show you this movie to give you a little idea about that. So here, this is a sunflower, some sunflower seedlings that are growing. And I just want you to observe their growth pattern in this movie. What they're doing here, this is sped up. Each frame is a 10-minute frame. So we're looking over, a couple of days here. I hope you can see that what they're doing what's called nutating. So they're spinning, the vine is spinning around and it's searching for a support. You saw right there, the vine managed to reach a support, and now it's changing its growth pattern to grow up and around the support. And the other guy's going to catch it right now. So you can see that plants, hopefully you get a sense that they're a little bit more reactive to their environment than you might have thought. But also you get a sense of sort of the time frame here. So the time scale of response to touch in a human being or an animal is very fast. The time scale of at least observable responses to touch in this plant is very slow because the response is a growth response. So it takes a little while to see it. So that's why I think a lot of people don't have an appreciation for plants moving. However, there are fast responses to touch in some types of plants. One example are these carnivorous plants. And one of my favorite carnivorous plant is this aquatic bladderwort called Utricularia gibba. And this is what it looks like. You can buy these actually at any store that sells stuff for your fish tanks. But what I want you to focus on is, I hope you can see my pointer here. I hope you can see these little sort of bladders here. If you zoom in on them, they look like this. And these are basically little tiny plant stomachs. So they have what's called an ultra-fast prey trapping mechanism. So there's a trap door right here that opens when a little swimming zooplankton, which you can see here, comes by. So here's a quick movie so you can see how this works. So there are trigger hairs on the surface of the trap door that sense the presence of the zooplankton and aspirate it in. So they're actually under suction, sort of as if you had sucked all the water out of a bladder, and then when the trap door opens, it sucks water in as the bladder opens. So these are really cool plants. They're actually related to snapdragons. And as I say, they trap zooplankton inside a bladder that's normally under negative pressure. The prey touch these trigger hairs, the trap door opens, and they're aspirated inside. And then they digest it. So this is a way for the plant to get special nutrients. Oops, okay. And then the last example I wanted to give you is, of course, the amazing mammalian ear. So in this case, we're animals or humans or mammals are responding to vibration. So sound waves enter your ear and they travel to the eardrum vibrates, and that vibration travels through these tiny bones in your middle ear. And this amplifies the sound vibrations and transmits them to these amazing inner ear hair cells, which I think you can see here. So each of these pillars here are a single cell, and they vibrate in response to the vibration. They move in response to the vibration, and that activates the release of neurotransmitters. And then, I don't really know, some stuff happens and you sense that there's sound. So I'm only interested in the perception part here. Okay, so I hope that I've convinced you that plants and animals need to sense mechanical force. And then the question is, how does that happen? And so the study of that question is called mechanotransduction, which there's a typo there, sorry about that. Mechanotransduction being the process by which a physical stimulus is transduced into a biochemical signal. So really, truly, the intersection of physics and biochemistry. Something physical that is a force that's out in the world becomes a biochemical signal that the cell can understand. So translating from the language of physics to the language of biology. And one way to think about this, is as a sort of subset of this of signal transduction. So one way that biologists often think about how cells sense and respond to their environment is through a process called signal transduction, where there's a signal outside the cell. It's perceived by a receptor, maybe in the plasma membrane or maybe intracellular. Those receptors then activate signal transduction pathways that lead to something downstream that changes cellular state in response to the signal. So the question, if you want to work under this paradigm, the question is, how do you do this when the signal is not a molecule? It's not a hormone, it's not a nutrient, it's not a morphogen, it's physics. It's a physical signal. And so trying to understand how this whole pathway is started from a physical process rather than a biochemical signal, that's exactly what my lab works on, and is exactly the question of mechanoperception. So the idea here is that there's a stimulus. So for us, we're interested in osmotic pressure or gravity, touch, shear force, vibration, etc. Oh, is there a question? Oh, no, that's from patients. Okay. Then at the end, at the end, you see a developmental response like we saw with the sunflower. You see tropic growth. Like we saw the sunflower growing around the stake, or there can be organ movement, like we just showed you with, like I just showed you with the utricularia, the carnivorous plant movement, or there can be a nerve impulse, as with the inner ear. But the big question is, how do you perceive this? And so the idea is that there are these specialized proteins or cellular structures or even tissues that change activity with applied force. So there's a stimulus, the mechanoreceptor senses the force, changes activity, and elicits the change in cellular state. One point I wanted to make is that these mechanoreceptors really can function at multiple scales. So there are entire organs that are designed to perceive the stimulus. So in the example of the bladderwort trapdoor, these trigger hairs are multicellular structures that lead to trapdoor opening when they sense the presence of the prey. There are also cellular mechanoreceptors. So people here in the CEMB have been studying how these trichomes, these are little hair cells on the surface of a leaf. They're sort of like little sensory organs that cover the leaves of plants, the leaves and stems of many plants. And when they are bent over, there's this purple here indicates that there's a calcium signal that then lets the plant know that there's probably an insect walking around on the leaf and it might be time to amp up its defense responses. What we're going to talk about for the rest of this lecture is the idea of molecular mechanoreceptors. So these are a great example here, are mechanosensitive ion channels, and that's what we're going to launch into. But before I do, well, actually, let me just ask, is there anybody have any questions they'd like to ask before I go any further? See a
Hi.
lot of, oh yeah, go ahead.
I just had a really naive question.
Sure.
So the video you presented at the beginning, like the plant and the vines and everything, how long was that? I know that each frame was like 10 minutes, but like the total time.
Yeah, I don't know the answer to that, but I would guess that was a couple of days. You saw it grew, probably this much. So under ideal conditions, that's probably a couple of days.
That was really cool. Okay,
I
thank
know, right? I know, right? Any other questions? Nope. Okay. All right, I'm going to go back and we're going to keep rocking this. Okay. All right. So before we launch into it, though, I just wanted to address what I'm sure you're all wondering, which is, plants and animals, they seem kind of different. Let's talk about that for a second. So plant cells, so I've illustrated here an example of hypocaudal cells. Hypocaudal is a specialized tissue that's really just connects the root cells to the shoot cells. They are surrounded by and also cemented together by a cell wall. It's made out of a material called cellulose. And you are going to be hearing a lot more about the cell wall later this week from Siobhan and Charlie. The cell wall gives the cell structure. It glues the cells together. So cell migration is not really much of a thing in plants. And it allows plants to build up what's called turgor pressure. I'll talk about that in a minute, but about 10 atmospheres. So you can think of each plant cell really as like a tiny little car tire that's under immense pressure and is providing the structure of the plant through turgor pressure. Turgor pressure of a plant cell is between 1 and 10 atmospheres, 0.1 to 1 megapascal. Now, if you look at your classic animal cell, here we've got a HeLa cell, those all came from Henrietta Lacks, and it's also surrounded by an extracellular matrix made of polysaccharide and proteins, but they do not have the same elastic modulus. And so cellular pressure in an animal cell is around 0.3 to 2 kilopascals. So we're talking 3 orders of magnitude difference between an animal cell and a plant cell. So, and as you know, plant cells can move around, animal cells can migrate. There's lots different between plants and animals. So keep that in mind as we go forward. Oh, and then this is just a little bit of explanation about turgor pressure and how that works. This facilitates plant cell expansion. You'll hear more about this. The cell wall is, when a plant cell needs to grow, it softens the cell wall, and that allows the cell to expand. That expansion can be driven or facilitated by turgor pressure. And the turgor pressure is created by this organelle here, this in blue, this is a vacuole. And so water will flow into the vacuole that will create osmotic pressure pressing the protoplast against the cell wall. So the cell wall is in black, the plasma membrane is here, and then this is the cytoplasm, and here's the vacuole. So the vacuole is pressing the contents of the protoplast out due to osmotic pressure, and the cell wall is constraining it in with strength, and that's basically what creates the turgor pressure. Okay, just something to think about. Okay, so here's the first think, pair, share exercise. So what I'm going to have you do, oops, I'm sorry. Gosh, this is hard to do. Okay, so what I want you to do is to list as many external, you might need to write this down because I think I'm going to have to stop sharing in order to put you into different groups. List as many external mechanical stimuli that plant and animal cells sense and respond to as you can. And I want you to think a little bit about ways in which plant and animal cells responding might be different and how they might be similar. So we're going to do this really quickly. I'm just going to give you 5 minutes to have a quick chat, and then we'll move forward. So let me see. Let me do this really quickly. Oh no. Oh, breakout rooms. Here we go. Okay. Sign into. Okay, there's going to be 3 or 4 of you. I'm just going to do some automatic assignment. So you're just going to find yourself in a breakout room, have a quick discussion, and we'll be back here in 5 minutes. Stephanie, there might be something you have to click. There you go.
Hey Liz, I would recommend pausing the recording because this is recording and you'll start recording.
recording again. What they say, if they hear what sounds like insects chewing. What else? Anybody else have something that's different, we think, between plants and animals with regard to mechanics and mechanoperception?
We talked about,
sorry, go
sorry.
ahead.
Okay. We talked about shear stress is really important in animal cells and human cells, but not necessarily. I don't know if that's as significant in plant cells.
Yeah,
and
it's
then we
hard to
talked
imagine how a plant cell would sense that.
Mhm.
Other than at sort of the whole tissue level. So there are like ocean plants that grow differently depending on how much the shear stress of the tides affect them. But at the single cell level, the way you look at it in animals, I don't think so. Fabiola, did you have something you wanted to say too?
So the one that I thought that it was like kind of different is just like light, because like plants need the light to survive. So they'll grow and try to face the light so they can do like photosynthesis and stuff. And us, like, we don't have that much of a response. Like, we don't necessarily need light to survive. So I guess that like that was the difference that I kind of thought that was pretty
Sure. Anybody else want to share before we zoom on? Okay, so I think the upshot here that I want you to take home from that is that actually plant and animal cells respond to almost everything mechanical, and perhaps the way they do it is different, but all of those physical signals that an animal cell needs to sense, a plant cell almost always needs to respond to it too, maybe in a different context, but they really are very similar in terms of the signals are very similar. Okay. Let's keep going on. Share screen. Let's keep cranking. And let me go here. All right. So we're going to talk about ion channels because this is the type of mechanoreceptor that we dearly love. So I'm going to tell you about that next. I want to talk a little bit about ion channels in general. Ion channels in general have a huge number of functions in plants, animals, and bacteria. They regulate osmotic stresses, ion homeostasis. As you know, they generate and transmit synaptic potentials, and as we're going to discuss, they're involved in mechanoresponses. So most channels are what's called gated, and that means that they rapidly switch between a conducting, that is an open state, and a non-conducting state in the presence of a stimulus. So here in green, you can see a closed state, a channel in its closed state. This is a voltage gated channel. You can see they're showing the transmembrane voltage here with plus and minus to show you there's a big transmembrane voltage. When this becomes, I guess they've reversed the polarity. I don't know when that happens, but they reverse the polarity of the membrane, and now the voltage gated channel is open and ions can pass across this little pore. Here's an example of a ligand gated channel, maybe a neurotransmitter binds like GABA or something, and that opens the channel. What we're interested in is this red class of channels that are tension gated. Okay. Mechanosensitive ion channels in general sense and respond to lateral membrane tension. So lateral membrane tension is tension that's in the plane of the membrane. So one way to think about this is you think about a cell. If we're talking about the plasma membrane, you think about the cell as a balloon, and you think about these ion channels that are all embedded in the balloon. If you blow up the balloon, like you add air to the balloon and it expands, that's increasing lateral tension in the membrane, in the material that makes up the balloon. And that lateral tension, that's what gates the channels. Once the channels open, ions can flow across them. So these are not transporters. They're not pumping, they're not using energy. They're just letting ions flow down their electrochemical gradient. So it could be in the cell or out of the cell. This can lead to a couple of different things. So if you have a cell, here I've shown in gray, and it swells. So maybe this is osmotic swelling, maybe it swells for another reason, but again, this causes the channel to gate. There's a couple things that could happen. If it's calcium permeable, calcium could enter the cell as you probably know, calcium is a second messenger, and it can work to activate all kinds of downstream effects. Or if it's anion selective, maybe it would let chloride ions exit the cell. In plants, this is exactly the direction that chloride would flow if an anion channel was open, and this would release osmolytes and could actually prevent the cell from expanding further. Or there are some cases where we think that mechanosensitive ion channels might have a non-conducting function or actually signal in ways that are separable from ion flux. But all of this is sort of based on this, what's called a two-state model for mechanosensitive ion channel opening. So you go from this closed state where ions cannot move to an open state where ions can flow. But this is pretty simplified, and really there are lots of other states. So something that you may have heard about is this idea of a desensitized channel. This is what happens to your inner ear when you go to a loud rock concert. And when you walk out of it, like you can't hear what anybody's saying, or you find yourself talking really loud. It's because the ion channels in your inner ear have become desensitized. They've actually closed. And for you to hear, you actually require more stimulation. There's also another conformation called inactivated. This is where the channel enters into a completely different conformation, and it essentially can't be opened until there's sort of a time period where it reverts to the closed state, and then it can be opened again. And this is just seems to be a way to just sort of stop if there is a stimulus that the organism wants a very transient response to. You get a transient opening, and then the channels basically close down and you can't respond any longer. It gives you a pulse of response. Okay, so how do we study this type of ion channel? In our lab, we use what's called single channel patch clamp electrophysiology, and it's a great way to study all kinds of biochemical characteristics and biophysical characteristics of a channel. So the way it works is you take a cell, we work in plants, but we express our channels often in Xenopus oocytes. They're these really big cells. They're super easy to work with. So we take a glass pipette. This is just depicting like a cross section through the tip of that glass pipette. We poke a little patch of the membrane, and so you sort of poke the pipette into the membrane. You sort of punch out a little disc of membrane so that it looks like this. This is like a cross section through, but you would have a disc of membrane inside the center of the glass pipette. And if you had, this is not to scale, by the way, if you had an ion channel embedded in the membrane, then you could add suction to the pipette, and you would deform the membrane like this. It would be adding lateral membrane tension, and you would gate the channel, and that would allow ions to pass. You would have we would have an electrode outside the pipette and electrode inside the pipette, and we would measure current across this tiny patch. So every time an ion opened, there'd be a little jump in current. So you can measure the opening stimulus, you could measure the amount of tension that you have to add or the amount of suction that you have to add to get the channel to open. You can measure how efficiently the ions pass and what kinds of ions pass across it. So there's lots of things that you can learn by doing this type of experiment. And in fact, this is the kind of experiment that was done wherein mechanosensitive ion channels were first discovered. And so I thought I would just talk to you a little bit about how these were first discovered way back in the 1990s, I think. So they took E. coli cells, and I should say at this time, people had just realized, this is 1990s, they just realized that bacteria have ion channels. For a long time, people thought that cells were too small to need ion channels. Turns out they do. So they take E. coli cells and they treat them with cephalexin, which allows them to divide but prevents the cells from separating after division. And you create these big, long snakes of many, many, many cells fused together at their ends. Add some lysozyme, which digests away the cell wall. E. coli have cell walls too, by the way. And these little snakes then inflate into what are called giant spheroplasts. And then you can patch them. Here's the patch pipette, just like I was talking about. And you can measure current across this tiny patch of membrane, or you can measure across the whole cell. So what happens when you do this is you apply suction within this patch clamp, and then you can create what's called a trace that looks like this, where the x-axis here is time, so this is during the time, and then the y-axis is current. So you can see the current is steady, and then you apply suction, and you get these stepwise increases in current. Each of those steps is one ion channel opening. So an ion channel opens, and then another one opens, and then another one opens, and then another. So it looks like they've got 4 channels in this patch. And then when they release the suction here, you can see those channels one by one closing. So it really is a single molecule imaging, but you're detecting the activity of individual channels in that pipette.
One question.
Yes, go ahead.
Wouldn't that process of making that huge cell change the sort of readings you're gonna get?
Yeah, I mean, that's a great question. It's just at the moment, the best mechanism we have for understanding is like physical access to the membrane. So for cell-walled organisms, there's like, that's what we have to do. And for animal cells, it's similar, right? People are patching cells that are growing in a monolayer on a glass slide. Like that's not actually how they normally grow, right? So yeah, so one of the big, I think, things the field needs right now is are some like biosensors for some of these processes. So we need to be able to look at membrane tension using a fluorescent marker dye or something that we can look at in vivo. We just don't have that at the moment. Any other questions? Okay. All right, that's a good question. Okay, so I'm just going to skip this part because I kind of talked about it already. But I did want to say, so electrophysiology is, like I said, one way to study some of these really basic things about how channels function, but there are all these other sort of ways to elicit mechanosensitive ion channel. We talked earlier about shear stress in A. With an animal cell, you can attach it to a surface and then stretch that surface. We're actually working in the CEMB to do this, to be able to do the same kind of experiment in plants. It's called plant on a chip, and you'll probably hear about that later. This is the electrophysiology that we do, where you pull a patch and you apply suction, but you can also monitor you can monitor current across the membrane when you like poke a cell with a glass rod. You can spray fluid. These are specific to hair cells, but from the inner ear, like I was telling you about, you can dislodge these hair cells with fluid or with a glass probe. Then you can also do osmotic swelling. And then there are even ways to grow animal cells on these little pillars that allow you can move the pillars with respect to each other and get like very tiny changes in substrate mechanics that you can then monitor how that affects mechanosensitive ion channels that are embedded in the membrane. So you can use ion-specific dyes, genetically encoded biosensors for, say, calcium, or electrophysiology to monitor ion flux in response to all of these types of stimuli. Okay, so I want to talk about a concept that's very near and dear to my heart, which we broached a little bit, which is this idea that mechanosensitive channels are ancient and universal. So what I'm showing you here is a very simplified and really at this point not all that accurate vision of the tree of life, with the origins of life here at the bottom, and then 3 of the main branches of life at the top. You'll notice archaea isn't really in here, but prokaryotes here, plants here, and animals here. And what I want to emphasize here in particular is that there were 2 points at which prokaryotes were endosymbiosed. So first, the endosymbiosis of the precursor of mitochondria here happened before plants and animals separated. So plants and animals are both eukaryotes. They have a common eukaryotic ancestor, and they inherited their mitochondria together. Then there was a second endosymbiosis in the plant lineage, and that created the chloroplast. So what's cool is that mechanosensitive ion channels are found in all of these branches. So I was telling you about these electrophysiology that was done in E. coli, and they discovered two really well-studied, the very first mechanosensitive ion channels to be discovered. They're called MISC-S and MISC-L. MISC-S stands for mechanosensitive channel of small conductance, and MISC-L stands for mechanosensitive channel of large conductance. Now, animals, we've identified a huge number of mechanosensitive channels. We don't find MISC-S and MISC-L there, but there are OSCAs, Piezos, TPKs, TRPs, TMC, and DEG-ENAC channels. There's probably more. There's probably papers in press right now with more new different kinds of channels from animals. Plants are cool because they have some of each of those. So plants have MISC-S-like channels, so channels that are related to these prokaryotic ones. These are the channels that my lab mostly works on. They also have some channels that are related to, that are homologous to animal channels, so OSCAs and Piezos. In fact, these OSCA channels, this family, this was first discovered in plants. And then there are some plant-specific channel families like the MCA channels. So there are some cool evolutionary history to study here. One thing that my lab is really interested in is how the piezo channels are functioning in plants. I can tell you about that offline sometime. But the thing to note here is that the concept of an ion channel serving to transmit changes in membrane tension to changes in ion flux is universal, but the environment in which those channels are functioning is not the same. Plants and prokaryotes have cell walls, animal cells don't. Plants and animals have intracellular organelles, and prokaryotes do not. So if you think just with a little plant-centric view here, you think about mechanosensitive ion channels and all the things, the range of things they could be doing. So there are lots of different families of ion channels, but they're also localized to a range of membranes. So I know this is like very sort of diagrammatic, but so I'll just sort of talk to you about what I'm trying to illustrate here. Here in gray, this is the cell wall, and the orange bar here is the plasma membrane. So there are some channels we know are in the plasma membrane in plant cells. MSL10 is a good example. The OSCA family members are probably there. These transport probably chloride ions and calcium ions respectively. But there are also channels found in the mitochondrial membrane. There are ion channels found in the plastid membrane, and there are ion channels found in the vacuolar membrane. We were talking about the vacuole earlier. That's the organelle that helps to create turgor pressure. And in fact, that's where we find piezo channels. Chances are good there are probably also ion channels in the ER and maybe even in the nucleus. So when we study these channels, we tend to study them individually. We just study MSL10. But the fact is, membrane tension is likely to affect a lot of other channels in the plasma membrane and potentially other channels inside the cell. And if you're changing chloride, you're changing the ion homeostasis within the cytoplasm, and you would likely affect all of these other channels. So it's very likely that there's a lot of crosstalk going on, not just between mechanosensitive ion channels, but also between mechanosensitive ion channels and other types of channels. Okay, I think I'm going to skip this and we'll do this part at the end, so we have make sure we have enough time, because I don't want to skip going into the biophysics of mechanosensitive ion channels. So, but let me pause. Does anybody have any questions just about the part I just covered? And then I'll talk about biophysics, and then we'll do the think, pair, share. Any questions or clarifications? Nope. Okay. All right. Well, I hope you have some questions about this next part, because it's definitely the coolest part. I don't know, coolest, but it's equally cool. So there's so the question, the biophysical question, the question for a biochemist, a biophysicist, is how do you get membrane tension to translate into opening an ion channel? So going back to the question I asked at the beginning of the lecture, how do you get a signal that is physical to activate a receptor? So there's lots of ideas, and really for a long time it seemed like there was like one mechanism for every mechanosensitive ion channel that was studied. But now things are starting to coalesce around a couple of different principles. So I'm going to discuss two of those. One is called hydrophobic mismatch, and one is lipid disordering. So hydrophobic mismatch. And the idea here is that mechanosensitive ion channels assume a conformation that, that's, I should say, that keeps the minimized lipid distortion. That's for the other one. They assume a conformation that keeps them embedded in the membrane. So if you think about a mechanosensitive ion channel with transmembrane helices that are normally embedded in the membrane, and then as you add tension, it actually thins the membrane. The membrane gets thinner as it gets stretched, and that can create, here in red, areas of the ion channel that are hydrophobic but are now exposed to the hydrophilic cytoplasm or extracellular space. And so what will happen, the ion channel will actually reorient in order to keep those transmembrane helices embedded in the membrane and restores matching to lipid protein contacts that have similar hydrophobicities. So one example of that is this ion channel I was just telling you about, the mechanosensitive channel of large conductance. So I think you can see here from this is like a cross section of the crystal structure in the closed state and in the open state. Here, these gray lines indicate where the membrane is. I think you can see that transmembrane tension here, moving from the closed state on the left to the open state on the right, causes these transmembrane helices to reorient. So if you just focus right here on this red helix here, you can see how it becomes more in the plane of the membrane as the membrane probably thins. So this is an interesting channel. It's a non-selective ion channel with a really big conductance, and is very well studied at the structural level. Okay. Right, so hydrophobic mismatch, as I said, likely contributes to its gating mechanism. So this is just a little bit more detail on how we think this works. So the idea is that when no tension is added, the hydrophobic zone here in red pretty much matches the width of the bilayer. But when you add tension to the membrane, now the hydrophobic zone has to be much narrower, and that energetic requirement favors the open conformation of the channel. Okay, does that make sense to folks? It's really just about keeping hydrophobic residues embedded in the membrane as the membrane becomes thinner under tension. You can think about blowing up a balloon, you know that the balloon material gets thinner. Okay, so the other idea is this idea of lipid disordering. And the idea is that a mechanosensitive ion channel takes a conformation that minimizes lipid distortion as long as there's enough free energy available. So the concept here, and I'm going to show you a couple of examples where we know this to be true, is that lipids are distorted around ion channels that are in their closed state. In fact, they tend to cause either a dimple, or here it's drawn as sort of, I guess you could think of this as a pimple. And once you add membrane tension to the system, you add energy to the system, and it allows the ion channel to assume a conformation that now reduces lipid disordering. And that's more favorable, and it also opens the channel. So I'll give you two examples of that. The first example is this channel Piezo. So this is a super, super cool and incredibly beautiful mechanosensitive ion channel from animals that's also found in plants. And in animals, it mediates mechanically activated calcium channels. It's involved in so many mechanoperceptive events, including touch perception, red blood cell volume, proprioception, which is like knowing where your limbs are even when you're not looking at them. It forms this beautiful trimeric propeller shape. So this top view, you're like looking down on the membrane. But I hope you can see here that the side view has an interesting sort of dimple look to it. And it's now been proposed that the gating mechanism actually involves flattening of this membrane dimple. So when it's closed, you can see here it's almost like it's like lying in a little dimple. And when you add tension, that flattens the dimple and allows the channel to then take the open conformation, and that's what opens it. In the case of Piezo, we actually don't have an open state structure. So this is pure conjecture about how it might open. In fact, there are lots of other models that people have put forward about how channel gating might happen. However, we just received notice today that this beautiful cryo-EM structure of MSL1 that we worked on with Peng Yuan's lab over at the med school provides evidence for exactly the same idea. So it's really cool. There's a channel from plant mitochondria that probably has the same gating mechanism as piezo channels from animals. So what you can see here in the bottom is the model that comes from the cryo-EM structure of MSL1. Here, up here where it says TM, this is the part that's embedded in the membrane. And I think you can see that these transmembrane helices, if you were to put a membrane in there, would create a dimple, very similar to what we see with MISC-S. Now we have open and closed state cryo-EM structures. So right, this movie is an animation imagining how the channel might move from one to the other. And I think you'll see what it implies is a flattening of that dimple. So I'm going to show you that movie right now. Am I? Yes. Okay. So now we're moving from, now we're in the open state. So this is the closed state structure where it's forming a membrane dimple. And then this is the open state structure where they're all flattening out. So oh, and then now you can see it from the top. As the channel opens, you can see the pore in the channel opening as those helices move flat. They go from sort of being perpendicular, partly perpendicular to the membrane, to being very flattened in the membrane, and they also flatten the membrane itself. So this basically argues that there may be some more universal mechanisms by which membrane tension can lead to channel gating, and that one of these is hydrophobic mismatch, and another one of those is probably this membrane flattening and area expansion. Okay, so with that, I just want to give you a little perspective, and then we're going to go back to that quick think, pair, share. So, or we can also have a question answer session. So what I wanted to talk to you a little bit about is this idea of membranes as being one of the most ancient platforms for sensing mechanical signals. So one of the most fundamental properties of a cell is its ownership of a lipid membrane. This is really what distinguishes what is life or what's inside. Sorry, I don't know why it's doing this automatic thing. What's alive and what's not alive. That's basically the definition of a cell, is the presence of a membrane. The biophysical environment of the membrane affects its function along with the function of any proteins embedded in it. And so these membrane properties are so universal that they underlie cellular and developmental processes in all organisms. And it has really been argued that osmotic force may be one of the original senses, and the ability to sense that one of the first sensations to have evolved. So, okay, so with that, with 5 minutes to go, I think I'll stop and see if you guys have any questions, and if not, we'll do a quick think, pair, share. What do you guys, any questions about anything I said or didn't say? Yeah, go ahead.
So it
Is it
seems
Gabe
that
or
Gabriel.
Gabriel?
Oh,
Yeah, go
Gabriel.
ahead. Yep.
It seems that the closed and open conformations of these channels it's like just the difference between a small hole and a big hole. So is it that they have like a charge density which keeps out ions from coming through, or how do they stop things from going through the small hole?
Right, so that's a huge area of controversy, and it depends on the channel. So sometimes that pore is too small to really even be wetted. So there's really no opportunity for any ions to flow through. They're not like sealed, but they're not, they're dewetted. And in other cases, I think that they're I don't think that I'm not sure that there's any evidence that there's a change in sort of the charge structure of the pore. Sort of what's lining the pore tends to still be the same residues. So you're not like swapping in and out charges. So I think it must really have to be about the pore size really is too small to fit water and ions through. Yep.
Okay.
Any other questions?
Question. So you mentioned that the channels are universal, but the mechanical environments are not.
Yeah.
So I guess with these channels, so the mechanical environment is tension. Is that different, or is it just the way that the tension is brought about is different?
Right, so that was sort of what I was hoping to get to in the think, pair, share. So
Okay.
think about a plasma membrane that is pressed against the cell wall with 10 atmospheres of pressure. That mechanical environment of compression is not present in an animal plasma membrane, and is also not present in like the vacuolar membrane or any of those other intracellular membranes. So compression is definitely one situation where the mechanical environment of a piezo channel in plant cells and a piezo channel in animal cells are different. Actually, that's why we think piezo channels are found in the vacuolar membrane of plant cells, is that's where they can function. Because that amount of compression probably affects the dimpling, right? I don't know if you can get dimples in a plasma membrane that's pressed against a cell wall with 10 atmospheres of pressure, but you probably can in the vacuole. That's our thinking.
I see what you mean. Okay,
Yep.
Thank you.
Any other thoughts? Questions?
I had a quick question. This is Ethan from WashU.
Hi, Ethan.
So you showed us those videos. That's got to be like, those things are tiny. How do you image those? How do you know that they look like that or that they behave like that? Or like what method did you use to find that out?
Are you talking about the ion channel video?
Mm-hmm.
Yeah, that's cryo-EM. So cryo-electron microscopy. So you purify the channels, you embed them in what are called lipid nanodiscs, and then you do electron microscopy at such high level that you can actually image the shape of the molecule. And then we did that in open state and closed state. So we introduced a mutation that creates a open state, and then we're guessing at how you get from one to the other. So we have an open state and a closed state, and then the transition between the two is our inference about how you get from one to the other.
It's really cool.
Thank you. All right, I think I got 2 minutes to go, so I'm going to let you guys. Oh, go ahead.
So Liz, here's the part where I might say maybe we take the opportunity to let them have a short break. I know you wanted to get to the take, pair, share. So
no, that's what I was going to say. I was going to say
okay.
Time for a natural break. Should I stop recording now?